Everyone has heard at least once that the climate is changing, that plastic is washing away into our oceans. But did you know it's estimated that humans have produced over 5.8 trillion kilograms of plastic waste since 1950, with nearly half of that occurring after 2004? That's within my lifetime, within our lifetimes. Can you imagine what 5.8 trillion kilograms looks like? For reference, the plastic water bottles dispensed by the dining hall weigh about 6 grams each. In my opinion, unmanaged waste is one of the most important issues facing our society today. My project explores the phylogeny of the IF's F64831 gene expressed in Ideonellus kinesis, a bacteria with innate plastic degrading enzyme activity. I chose to research this topic because of what its scientific advancement could mean for the Earth and its environment. The ISF64831 gene found in Ideonellus kinesis bacteria. This bacteria was only just discovered in 2016 near a plastic recycling facility in Japan. It had been growing on microfilaments of polyethylene terephthalate, or PET for short, which is a plastic similar to polyester in nature and applications. By use of its two hydrolyzing enzymes, PETase and MHETase, the bacteria is able to turn plastic into the major carbon and energy sources of the cell. The breakdown of PET can be described in 3 steps. First, PETase catalyzes the hydrolysis of the ester bonds present between terephthalic monomers, producing monoterephthalic acid, otherwise known as MHET. Then, MHETase causes another ester hydrolysis to occur, generating 2, what we would call environmentally benign molecules, terephthalic acid, or TPA, and ethylene glycol. Finally, TPA is oxidized via TPA dioxygenase, and the whole process concludes in the release of waste carbon dioxide. The research of this project is centered around the hypothesis: if Ideonellus kinesis and various other bacteria evolved from a common ancestor, then those bacteria may also contain similar gene sequences, potentially allowing them to express plastic-degrading enzymes. Using the BLAST database, we bioinformatically compared the gene sequence of Ideonellus kinesis to that of all other known bacteria. The relationships between the DNA of these prokaryotic life forms serves as the base upon which our phylogenetic tree was generated. This tree shows the evolution of the gene and the related organisms. The percent similarity between the gene sequences is represented a little more clearly here on this graph. You can see that the organisms whose gene sequence most closely resembles IFF64831 is that of Acidivorax staphylides bacteria, with close to 84% gene sequence similarity. A closer look at their gene sequences reveals just how similar they truly are. This figure places the two bacteria side by side to allow for an easy comparison. You see that large chunks of the gene sequence are identical, with slight variations occurring throughout. However, one major difference is in the number of disulfide bonds. So you can see the additional disulfide bond found only in Ideonellus kinesis's PETase enzyme is underlined in green. As previously discussed, we've determined that the closest relative of this gene is the PBSA gene expressed in Acidivorax staphylides bacteria. PBSA encodes an enzyme, polyteramethylene cysteine depolymerase, that breaks down PBS and other biodegradable plastics. Additional evidence of common evolution in these two bacteria can be found by examining their catalytic mechanisms. In Ideonellus kinesis's PETase, the catalytic triad is composed of serine 160, aspartate 206, and histine 237. In PBSA, the serine hist asp triad is conserved. Both enzymes contain similar protein structure, disulfide bonds, beta sheets, alpha helixes, etc. In addition, both belong to the order Burkholderalides, a classification of probacteria. When examined together, this information confirms that Ideonellus kinesis and Acidivorax staphylides bacteria, both having evolved from Polyangium bransporum, share a highly similar gene sequence. This similarity allows them the ability to degrade different molecules of plastic. The study of IFF64831 gene and its phylogeny excites scientists to discover better, faster, and more environmentally conscientious ways of recycling, bringing humanity one step closer to solving the world's plastic pandemic. With new insights in the field of plastic degradation discovered daily, perhaps the entire world will soon have the resources to not only think, but act 80-20 as we strive to here at Deerfield. Thank you.